Hello everyone, a very good morning to all. In my last lecture, I was completed active transport and passive transport. So, welcome to raw biology classes guys. Today we starts bulk transport. This is also the type of membrane transport. So, with this, I will completed membrane transport process today. So, let's start this lecture without wasting time. Bulk transport. Here, bulk means that in large amount. Okay, transport means that movement. So, bulk transport we defined as transport of substances in bulk form across the plasma membrane. Okay, large amount में substances plasma membrane के across ट्रांसफर करे या मूवमेंट उन substances की सो उस प्रोसेस या उस ट्रांसपोर्ट को हम बोलेंगे बल्क ट्रांसपोर्ट. ओके, सो लेट्स define transport of substances across plasma membrane in bulk form. ओके, सब्सटेंसेस का ट्रांसपोर्ट होना अक्रॉस प्लाज्मा मेम्ब्रेन या सब्सटेंसेस का मूवमेंट अक्रॉस प्लाज्मा मेम्ब्रेन इन बल्क फॉर्म. ये सब्सटेंसेस सॉलिड भी हो सकते हैं एंड लिक्विड भी हो सकते हैं. On the basis of this type of medium, whether the substance becomes solid or liquid, I was discuss the type of transport occur later. So, first the bulk transport process is further divided into two methods, or it became occur by two methods. It became an endocytosis or through exocytosis process. So, let's discuss guys. Bulk transport became endocytosis process and exocytosis process. Okay, first we discuss in detail endocytosis process. Okay, endo means that within the cell. Okay, within cell. Okay, within. Cytosis means that cell. So, inward transfer of any particles such as food, pathogen and other substances by forming carrier vessels or by means of carrier vessel. This type of transport we say that endocytosis. Okay, so simply in simple language, जो इनवर्ड मूवमेंट होती है किसी भी सब्सटेंस की अक्रॉस प्लाज्मा मेम्ब्रेन बाय कैरियर वेसल्स की हेल्प से, बाय फॉर्मिंग और बाय मींस ऑफ कैरियर वेसल्स की हेल्प से सेल के अंदर इनवर्ड मूवमेंट होना किसी भी सब्सटेंसेस की, जो सब्सटेंस सेल को चाहिए एक्स्ट्रा सेलुलर स्पेस से उनकी इनवर्ड मूवमेंट होना सेल के साइटोप्लाज्म के अंदर, उस प्रोसेस को हम एंडोसाइटोसिस प्रोसेस बोलेंगे. फर्दर, एक्सो मींस दैट आउटसाइड. ओके, सेल means that the outward movement of substances across the plasma membrane. Here जो substances सेल को रिक्वायरमेंट नहीं है जिन substances की, या हम बोल सकते हैं undigested food for example हमारी बॉडी के अंदर जो भी होता है उनका मूवमेंट आउटसाइड द सेल, उस मूवमेंट को हम बोल सकते हैं या ट्रांसपोर्ट को बोल सकते हैं एक्सोसाइटोसिस. सो लेट्स डिस्कस एंडोसाइटोसिस प्रोसेस इन डिटेल. Here, the exocytosis, we also call that cell vomiting process. Okay. So, let's discuss endocytosis first of all. As you already know that endocytosis is the inward movement of substances plasma membrane by means of carrier vessels. By means of carrier vesicles. Okay, so let's discuss endocytosis. For that, the endocytosis process, guys, performed by two methods. It became a phagocytosis or it became a pinocytosis. Phago means that to eat, or cytosis means that simply cell. So, cell eating process. We commonly call that phagocytosis is a cell eating process. Where pinocytosis means that, pino means that to drink, a cytosis means that cell. So we simply said that pinocytosis is also known as cell drinking process. So let's discuss, guys, phagocytosis and pinocytosis. These are in your syllabus. So let's discuss this topic. Here, phagocytosis. Phago means that to eat, or simply cytosis means that cell. So we call that it is a cell eating process. Cell eating process. Okay, but there is an important difference is here is that pinocytosis means that cell drinking, or here is the cell eating process. So in phagocytosis, solid particles are transferred across the plasma membrane. But in pinocytosis, here dissolved substances or the fluid matter are transported across the plasma membrane. So in phagocytosis, in phagocytosis, we discussed that it is a cell eating process and we define as movement of solid particles such as food, pathogen, etc. across plasma membrane by means of detachable vesicles. Here, detachable vessels is also known as carrier vessels, and in phagocytosis, later we call that phagosomes. Okay, these vessels we call that phagosomes inside the cell. Okay, the movement is occur by when the invagellation of plasma membrane, due to invagellation of plasma membrane across the substances which require the cell to take from extracellular to intracellular. Okay, so through diagrammatically we discuss that suppose this is a cell, here this is a membrane. Okay, so there is a solid substances present outside this is an extracellular space. Extracellular space, or this is intracellular. Space. Okay, when the cell required any substances from extracellular, the plasma membrane start engulfing it. Okay, they start folding across the substances. सो सेकेंडली इसे हम छोटा बना देते हैं. ओके. सेकेंडली जो प्लाज्मा मेम्ब्रेन है वो कंप्लीटली क्लोज कर लेगी उस सब्सटेंस को, एंड फर्दर ये जो है वो वेसल्स के फॉर्म में वेसल बना लेगी एंड डिटैच हो जाएगी प्लाज्मा मेम्ब्रेन से. डिटैच होने के बाद इसको हम क्या बोलेंगे? फैगोसोम. फैगोसोम inside the cytoplasm. Further, this phagosome contains digestible substances, so they fuse with lysosome, which present inside the cell or cell cytoplasm. When they fuse with lysosome, we call that lysophago. This vessel we call that lysophagosome. Okay. So, and one thing is that the phagosome, here the phagosome was in 1 or 2 micrometer in size. Okay. Further, these phagosomes when these lysosome phagosomes form, here the digestible substance are digested and fuse into the cytoplasm, and the undigested substances remain into the vessel. Here this vessel we call that residual vessel. Residual vesicle. Okay. So, further this residual vesicle is move outside the cell. Okay. Outside the cell because they contain undigestible substance. So, this outward movement of undigestible substances through the plasma membrane, we call that exocytosis or cell vomiting process. Okay. So, it is a very simple process. Let's discuss stepwise. Phagocytosis मैंने आपको बताया, it is a cell eating process है. बट इसमें मेनली क्या है? सेल ईटिंग है, तो मीन्स दैट सॉलिड पार्टिकल्स ही सेल के अंदर इस प्रोसेस के थ्रू एंटर करेंगे. सो व्हेन सॉलिड पार्टिकल सेल्स रिक्वायर एनी सॉलिड पार्टिकल, दे स्टार्ट फॉर्मिंग वेसल्स अराउंड द सब्सटेंस और दे स्टार्ट देयर प्लाज्मा मेम्ब्रेन इन अराउंड दिस फोल्डिंग. ओके. सो when they cover the whole substance, they form a vessels, detachable vessels, which we call that phagosomes, whose diameter is 1 to micrometer. Further, when they enter into cytoplasm, they fuse with lysosome. These lysosomes contain digestible enzymes, okay, which were required to digest the substances. Further, when the lysosome fuse with this phagosome, they form phagolysosomes, okay. So, here the substances which are digestible are digested and fuse into the cytoplasm. But the substances which are undigestible are remain in this vessel, or here this vessel with residual substances, we call that residual vesicles. Okay. Then further, these residual vesicles are transferred outside the cell or move outside the cell. This movement we call that exocytosis or cell vomiting process. Okay, this we simply call that phagocytosis. All about food intake, solid food intake. Here the last one is pinocytosis. It is not so different about in comparison to phagocytosis. Here simply where the vessels we call that phagosomes, here the vesicles are pinosomes. Okay, but here the main difference is that they take fluid matter and dissolved substances. So, This process we define as that movement of fluid matter and dissolving substances across the plasma membrane by means of carrier vessels, or we call that detachable vessels. These vessels further we call that pinosomes. Here the pinosome size is 100 to 200 nanometer. Okay, 100 to 200 nanometer. When the pinosomes entered into the cytoplasm, when they contain digestible material, they fuse with lysosomes. But when they do not contain any digestible material, they simply diffuse into the cytoplasm. Okay, but the solute which transferred from this, they became selective or non-selective. They become selective or non-selective. When the selective solute are transferred through this process, these ligands are combined with the receptor site which are present in plasma membrane, and they form a complex. Further, they move into the cytoplasm. Here, they dissolve or fuse into the cytoplasm. Okay, this simply is a pinocyte process. So, with this, here the membrane transport process is complete. So, guys, the membrane transport, a simple discussion about whole membrane transport process is that it became three type: active, passive and blood transport. But here the active transport are energy dependent process, but passive transport is energy independent. But the bulk transport is also not surely an energy dependent or energy independent. The endocytosis process here energy dependent process. They requires energy. Okay. So let's complete this complete this topic today. Let's start the next topic in my next video. So please guys, like, share and subscribe my channel. Thank you so much for listening me.